Say, have you thought about curing diseases, resolving the food crisis, protecting endangered species? Well, all that may just be possible thanks to genome editing, more specifically, CRISPR. But to understand that, let's dig a little deeper. Let's talk about genes, which is the sequence of bases on DNA that determine traits that an individual has. They are specific and unique for each trait. Suppose this gene is responsible for hair color.
A few mutations later.
Now let's get back to how CRISPR works. It employs Cas9 mechanism which involves DNA endonuclease enzyme fitted with a guide RNA. Imagine a blind person with a guide dog. The guide RNA snips out the bit of DNA which is complementary to it, and the enzyme goes snip snip and splits the DNA into two. The cell now goes into panic mode and tries to desperately repair the DNA. In one process, it just joins the end like when you break your sister's lipstick and put it together before she notices.
Shaika!
It was so cute! In the other one, it uses the DNA of its homologous chromosome to fill in the gap. We explored the latter process. We manufacture DNA which is same on either side, except it has a modified gene that gives healthier or improved characteristics, example hardier crops. And we get our required gene. We can now have disease-resistant plants, treat human diseases, get rid of pathogenic microorganisms, and so much more. Now let's talk about why CRISPR is better than alternatives such as Talon or zinc finger. It's easier and cheaper to manufacture guide RNAs to edit DNA, and it's more efficient and less time consuming as we don't require too many proteins to edit different segments, as we could simply add more guide RNAs. Now let's look into some of the possible disadvantages. Uncertainty in the long run arises because we need to use vectors such as viruses for editing genes in multicellular organisms, and we don't know the harmful effects of these viruses staying in the body for too long. Then there's genes and environment. The interaction between the two causes traits to be expressed, so unless we know all the responsible genes and possible environmental triggers, we can't effectively treat anything. Lastly, there's a heated debate of whether creating perfectly healthy individuals by manipulating embryo DNA is ethical or not. The most fascinating thing about CRISPR is it's not only a marvelous breakthrough, it's also a philosophical turning point in human history. If executed wisely, we could revolutionize the whole world and all living creatures in it, using just tiny nitrogenous bases.